Conjugation is the contact between donor and recipient cell, and this is mediated by the sex villi or the villi or F villi, which are made by only F plus 10. This villi acts as the attachment site for the F minus cells. So, the in A plus cells, these villi are present. I already told you A plus cell, why these A plus cells are called as the A plus, because these cells are having the A plasmid. This plasmid is called as the A plasmid fertility factor. And if one copy is present, one villi is present. Okay. Hence, these A plus cells are called these donor cells are called as the A plus. F minus cell does not contains the F factor F villi. Hence, the F minus cells have the receptors on their cell wall, on their surface, cell surface, receptors for the villi. When the F plus, that is the donor, when F plus E. coli comes in contact with the F minus E. coli, then a attachment in between these two cells takes place by means of the presence of villi. Okay. Okay. So transfer of the A plasmid takes place from A plus cell to F minus cell. From that junction where the two cell membranes are attached, not from the uh, villi where the villi it is attached to the F minus cell. So that this always remember, the transfer of the plasmid never occurs through the villi, it only occurs through the junction which is made in between both the cells. So pillin is polymerized into the two hollow cylinder of the 8 nanometer diameter and 2 nanometer axial hole. The sex pilus is necessary for contact in between donor and recipient cell. It is only important only for the contact in between the donor cell and recipient cell. When the tip of a pilus contacts with the surface of recipient cell, then mating is initiated, and mating cannot occur between two A plus cells because the genes TRA-S and TRA-D codes for the surface exclusion protein. The TRA-Y and TRA-I makes the nick in the one strand of the F factor at the point origin of the transfer, and from that transfer of the this A plasmid starts. Then this protein complex with the TRA-Y and TRA-I and binds to the DNA and causes the unwinding of the DNA. This protein complex moves along with the DNA. Okay, this protein may also called as the relaxase protein. Okay, relaxase. This the 5 prime end of the cut end enters into the recipient cell and is released when circle gets back into the origin. So that the when complete transfer of this plasmid occurs from A plus cell to the F minus cell, then this mating further uh, resolves between two cells. So that this transfer, how this transfer occurs. So the William Hayes in 1953 demonstrated the genetic exchange in E. coli. In between the genetic transfer, it is made in F plus cell. Okay, F is the self-replicating circular plasmid. Okay, A plasmid contains the origin of the transfer definitely, where the initiation of the transfer starts. Now, actual process we are going to see. This is the conjugation process. Okay, and here you can see this electron micrograph. This is the A plus cell and this is F minus cell, and their attachment took place here. Okay, so you can see transfer. By means of the pili, these two cells are get joined and conjugation bridge is formed. But this portion of the cell membrane in between two cells also uh, mix together, and junction is formed in between the cell membrane and transfer thereof, not from the dish tube. It is only diagrammatically shown, but transfer never takes place through the conjugation bridge. Okay, so this conjugation between A plus and A minus cell, this is the A plus cell. This blue color is the bacterial chromosome. This red color is the F factor or A plasmid. Okay, hence it is the A plus cell. This F minus cell does not have this plasmid F factor. Okay, hence it is called as the F minus cell. This F plasmid, it is nicked at the origin of the transfer by maybe the YZ nucleases, which are again encoded by the plasmid itself. Okay, and this plasmid transfer uh, where the nick is made, and the relaxase protein attaches to this end, or this particular YZ nucleases remains attached, and it movement of or transfer of this A plasmid takes place from donor cell to the recipient cell. And this transfer, you can see the it is extended further, and this transfer at the same time replication of this DNA starts. Okay, and transfer of DNA when completes, then definitely in host cell also, and in the F minus cell also, the replication completes there. And this F minus cell initially it is get converted to A plus because it also gets the one copy of the plasmid. This A plus cell always remains A plus. It never get converted into from A plus to F minus. Oh, because only one strand of the A plasmid, transfer of the only one strand of the A plasmid takes place into the F minus cell. Always remember. The result of conjugation is that the recipient cell acquires the F factor. Thus, convert this cell converted from F minus cell to F plus cell. F plus cell remains unchanged. In some cases, F factor may carry genes that were once found onto the bacterial chromosome. These types of the F factors are called as the F prime factors. That initially we discussed about F prime. So diagrammatically, we